As Jerry mentioned, I'm going to be telling you a little bit about the PhD work that I did at the University of Saskatchewan in the Department of Chemistry, working with Graham George and Ingrid Pickering. But I'm now a postdoctoral fellow at Johns Hopkins, working with Svetlana Litsenko. And so today I'm going to talk to you about X-ray absorption spectroscopy in understanding and treating Alzheimer's disease. They advanced a minute ago. There we go. So I'll begin by telling you a little bit about neurodegenerative protein misfolding diseases. A number of these you've most likely heard of. These include diseases like Alzheimer's disease, Parkinson's disease, Creutzfeldt-Jakob disease, or more commonly known as mad cow disease, as well as Huntington's disease. And what these diseases have in common is that they all have proteins that misfold and contribute in some way to the disease pathology. And once these peptides have aggregated, they can form fibrils or filaments, and in this case, within the brain. And today I'm going to be focusing on Alzheimer's disease and one of the misfolded proteins called amyloid beta, which forms amyloid fibrils and eventually can form amyloid plaques within the brain. Now, there are actually 35.5 million people worldwide with dementia, with Alzheimer's disease being the single most common cause of dementia. It's actually the 5th leading cause of death worldwide, and it kills more people each year than breast and prostate cancer combined. And unfortunately, Alzheimer's disease is the only cause of death in the top 10 in North America that cannot be prevented, cured, or really slowed. And one of the major questions in Alzheimer's disease has been, what is actually a cause or an effect of the disease? One of the major observations is this brain atrophy or wasting that occurs in Alzheimer's disease. And you can see on the left-hand side here in gray, this is just a cartoon of the human brain. And so on the left is the healthy brain tissue with a few key areas labeled just to orient. You can see the cerebral cortex, the ventricles, and the hippocampus. On the right-hand side, you can see what happens to the brain in severe Alzheimer's disease. So you can kind of see the shadow of the healthy brain tissue behind, and then the Alzheimer's tissue is just colored green. And there's significant atrophy of the cerebral cortex as well as the hippocampus, and the ventricles are significantly enlarged. On a more microscopic scale, the misfolded proteins that I mentioned, like amyloid beta, actually accumulate as plaques in areas of the brain that are important for memory and cognition. So these include the hippocampus primarily. So just to show a bit of a schematic in one hypothesis of how the aggregation of A beta might occur. So here in this cartoon, we're looking at a healthy synapse. And I'll just turn on the laser pointer. So this is in the brain. These are the neurites, just to orient everybody. And typically as part of the signal, metal ions such as zinc and copper are released within this synapse. And these create a number of different signals. Any excess zinc or copper is typically stored in proteins like metallothionein. They also go on to signal within the cell. Amyloid beta can be present in the synapse, but under normal conditions, any A beta peptide is degraded. In Alzheimer's disease, also looking at a similar synapse, we have a lot more zinc and copper ions within this synapse. These ions are not stored in metallothionein, and they don't seem to go on to trigger the same signaling pathways within neurons. And to make matters worse, the amyloid beta that's normally degraded is not, setting up the perfect environment for it to interact with these excess zinc and copper ions. This triggers the formation of amyloid beta oligomers and can go on to form these larger amyloid beta plaques. And this, of course, is just one hypothesis as to how this can happen. But based on this hypothesis, a number of studies have looked at metal chelation therapy as a way to fix some of the problems that are happening here. And one of the compounds that people have looked at are the family of drugs called 8-hydroxyquinoline compounds. And here's just the basic 8-hydroxyquinoline compound here. And in the cartoon, I just wanted to show you that it's sort of this starburst shape in our cartoon that we're sticking with. And 8-hydroxyquinolines are generally thought to work as metal ion chelators. And in this situation, these chelators are able to bind some of the excess zinc and copper within the synapse. And it was thought that they would bind and mobilize metals from both the soluble amyloid beta and the oligomers, as well as the plaques. And this would trigger pathways. Storage of metals will be turned back on, signaling pathways will be turned back on, and hopefully, and maybe most importantly, we would be able to restore metals to the surrounding neurons and upregulate proteases that would degrade amyloid plaques. And there was actually some really early evidence that provided some strong support for this hypothesis. In mouse models, 8-hydroxyquinoline treatment shows a really striking plaque clearance. So on the left here in an Alzheimer's model mouse brain, all of these little black spots are stains for amyloid beta. And you can see after the Alzheimer's mouse has been treated with one of these 8-hydroxyquinolines called PBT2, there's significantly less of these dark spots within the brain. And so to show some further evidence here, this is a study looking at Alzheimer's model mouse. In the untreated mouse, you can see that he has this black spot sort of swimming around in this pool. And he would have been trained to swim to this is a platform here where he can get up out of the water. And unfortunately, you can see him swimming around and he's forgotten what he's supposed to do or forgotten how to get up out of the water and up to this platform. And you can see him sort of swim by a couple of times without success. And so you can really see how in the Alzheimer's model, how the progression of the disease affects the memory and cognition of the animal. And I can start the second video here. In the PBT2 treated mouse, you can see how he very quickly swims directly across to the platform and is able to get up out of the water. And this is just a really striking example of what was seen in the mouse model in some of these early studies. And so this was really what led us to develop some of our research questions for my PhD. So we wanted to look at how does amyloid beta bind copper-2 in solution? How do 8-hydroxyquinolines bind copper in solution? And then how might 8-hydroxyquinolines interact with amyloid beta, also in solution? But before I jump right into the exciting results, I, of course, will just briefly talk about the methods. So, of course, we're using X-ray absorption spectroscopy. All of my data was collected on solution samples. In many cases, these are dilute samples. These are purified proteins in solution or small molecules. It was an excellent technique for our purposes because it required very little sample prep. So once you have your purified protein, these are just simply in buffers and then flash frozen, similar with the small molecules. And it allowed us to get very detailed structural information. We typically couple X-ray absorption spectroscopy with computational chemistry. specifically density functional theory, and we use this to predict our structures. And this is typically considered a theoretical, but it can be a very rigorous approach. And I just wanted to clarify some kind of nomenclature. I know there's a few different ways that people talk about the same thing within the field, unfortunately. So I just want to show you what we use. So we describe this region as the near edge, and of course, this region as the x-axis. And so if we just zoom in a little bit, we can kind of divide it into 3 components. And so the near edge spectrum on the left in the sort of green color, the near edge spectrum can give us information on the electronic structure, the oxidation state, can tell us a little bit about the coordination geometry and the type of ligands. Whereas the x-axis, which I wanted to note that we typically k-cube our data, our x-axis data. So in all the examples that I show will be k-cubed. This can give us very accurate metal ligand bond distances. It can also give us information on the type of ligands, as well as the number of ligands at a specific distance. But this requires fitting a model, which is where our computational chemistry comes in. And then, of course, there is the x-axis Fourier transform, which is arguably a really nice sort of visual representation of the x-axis. And you can think of it as the copper, in this case, being at 0. And the first shell ligands, in this case, being about 2.5. And then we have some outer shell ligands around 4 and 5 angstroms here. And to just give a couple of examples of a near edge spectrum and highlight some of the distinctions that it can make, you can see on the left is a copper 1 spectrum. And in copper 1 spectra, one of the most prominent transitions is this 1s to 4p. And it can give us a little bit of information about the coordination of the copper 1. This peak is typically very high in digonal complexes. This, I believe, is an example of a trigonal complex, and it's often a bit smaller in a tetrahedral geometry. On the right-hand side is an example of a copper 2 spectrum. In this case, there are fewer sort of sharp, distinctive peaks typically in the edge. One of the key components is the 1s to 3d transition, which is actually really small down here. You kind of have to take my word for it in some of these spectra, as well as the main edge here is the 1s to 4p transitions as well. And briefly, I wanted to introduce a technique that we've been using more and more as it gains sort of in popularity. It's called high energy resolution fluorescence detected, or HERFD XAS. And as you can see, these, I won't go into the physics so much about it, but it's significantly improved resolution over the conventional XAS. So the solid line is the HERFD spectrum, and the conventional XAS is now the dashed line. You can see how much sharper the 1s to 4p is in the copper 1 spectrum. And you don't actually have to take my word for it anymore. You can actually see that there's a 1s to 3d transition down here. And you can also see that there are other peaks that are more well-defined in the HERFD spectrum. And with our computational chemistry, this is really how we build our x-axis model. Through density functional theory, which is considered the gold standard for transition metal complexes, because it can accurately predict the structures. And these are energy minimized, or in this case, enthalpies. This is a theoretical but rigorous method, as I mentioned. And this is really what provides our model for simulating our x-axis. And so in the image on the right-hand side, I've just shown a few different copper coordination like possible conformations in the same molecule. And so we can model these, find our sort of most favorable structure, and then use that to simulate our x-axis. We typically do this using a program called FEF9. It's an ab initio multiple scattering code that I believe is publicly available online. We also use a program called OPT to fit our simulated XAFs to our experimental data. OPT is a component of the XAFs Pack suite of programs, which is written by my PhD supervisors and also available online at this link down here. And so we use OPT for our data reduction and analyses. The fitting gives us corrected interatomic distances, and we can actually link the variables so that we preserve our overall calculated structure. And so without further ado, into the results and our first question of how does amyloid beta bind copper in solution? It's fairly well established that it's likely 4 or 5 coordinate copper. Also emerged in the last, more recently, I guess, not the last few years, but that there are 2 coordination environments that depend on pH. There's a low pH component and a higher pH component, so that it's actually a mixture at more biological pH of about 7.4. It's also fairly well agreed that the ligands likely include histidine. There are 3 histidines at the N terminus in the 6th, 13th, and 14th positions. However, the other ligands there's less agreement about what those might be. Some studies have proposed the N terminus, others have proposed tyrosine, aspartate, alanine, and even exogenous molecules like water. And unfortunately, investigations into the copper binding site in A beta have been complicated for a number of reasons. The major one is that it's very difficult to study the full-length peptide because it has this tendency to oligomerize and aggregate. There are also problems with heterogeneity. As I mentioned, there are a couple of different components that exist at different pH, so that, of course, complicates things. And photoreduction is also a big one, specific to X-ray absorption spectroscopy. And so I just want to kind of illustrate what happens. And this is actually an experiment that we had done. So if you just look on the left-hand panel, the green is actually our first sweep of our sample. So when I say sweep, I mean sweep in energy. And you can see that after several hours in the beam, in this case, our last sweep, you can see that there's quite a big change. This is actually the copper 1s to 4p transition coming in here. And you can see that over time, after about 7 hours in the beam or so, we extrapolate that there will be almost entirely copper 1 in our sample. And this is actually really striking because these samples are frozen at liquid helium temperatures. And so we wanted to, not to pick on anyone, but to illustrate our point that this has been an issue in the literature. The dashed line here is our initial first sweep, and now the solid line here is our final sweep. And this red spectrum here is actually one that we borrowed from an older paper in the literature, when photoreduction was less well known and just not thought about as much. So this, of course, led to some difficulty interpreting the data in some of the early studies. And so with our work, we used a beamline at SSRL that has a really big beam. It's really perfect for doing this kind of work because it has a lower flux density, and we're able to move the sample, and each sweep uses fresh piece of sample to hopefully as much copper 2 in our sample as possible at the end of the sweep. This is a lot more difficult on beamlines where they have a really small spot, really high flux density. We actually did a few experiments at the Australian synchrotron where they have such really high flux on their XIS beamline that we had lost most of our copper 2 before we got to the edge of the first sweep. So it can be a real challenge. I also wanted to mention that there is very strong evidence for histidine coordination of the copper 2 in amyloid beta. in actually plaque cores that were isolated from human brain tissue, copper-2 was found to be bound to amyloid beta via histidines. This is a relatively old study now that used Raman. In more recent studies, we've gotten a little bit more creative. There's isotopically labeled amyloid beta. They've used targeted amyloid beta mutants, excuse me, a range of solution pH, as well as advanced spectroscopies to suggest that actually these 3 histidine residues might rapidly exchange in solution. So that most of the time you have histidine 13, histidine 14 bound, but histidine 6 may be bound part of the time as well. And so we started out by collecting spectra of copper-2 amyloid beta 1 to 42. So this is the full-length peptide at 3 different pH values. We chose pH 6.1, 7.4, and pH 9. And these are the conventional XAS spectra. And as you can see, right away, there's a fairly distinct peak at pH 6 that's not as visible at either of the other 2 pH values. And so this seemed to suggest to us that they may have different geometry. And so, of course, we collected some HERFD XAS, and in the HERFD spectra, it's a lot easier to see that they're very distinctive peaks within the near edge. And so this really convinced us that we likely had a different geometry at not just the 2 different pH values, but probably at all 3. Looking at the XAFs, and now I've switched things so that our data is shown as a solid line, and the fit is now shown in the dashed line. These are, this is from the conventional XAS. This is not HERFD. And you can see that there are different backscattering contributions at the 3 different pH values, just looking at this first shell here. Similarly, looking at the Fourier transform, we concluded from looking at this data that there is a mixture of copper-2 coordination environments, likely at all 3 pH values tested. And so we went on to focus on this region, which is commonly accepted to be the area where we would find multiple scattering to histidine residues. And our purpose of doing this was to see if we could get a better idea of how many histidines might be bound in our samples. And of course, although the imidazole ring looks fairly simple, it actually leads to multiple single and multiple scattering paths. So you of course have your copper nitrogen path, and I've included copper oxygen path here to complete our coordination number. We also have paths to carbons 2 and 3 here. And these distances can be affected by kind of the angle of the imidazole ring. It can actually tilt sort of side to side in solution, and it can also tilt sort of up and down. And this will affect these distances as well as the scattering from the copper to this carbon 4 and this nitrogen labeled 5 here. We also have multiple scattering from the copper through the nitrogen to the carbon 4 and to the nitrogen 5. And you can see how each of those paths combine into our fit, which is the dashed line. Similarly, just to show the Fourier transform, you can see how these first shell distances combine, as well as how all of those multiple scattering paths combine to fit our histidines. And so through lots of fitting and going back and forth and testing different models, we came to the conclusion that we do have 3 different copper binding sites in amyloid beta, or I shouldn't say different sites, but different conformations at the 3 different pH values. So the orange spectrum is a pH 6.1. And in this case, you can see the region where the histidine multiple scattering is observed is relatively small. And so we believe that we have just one single histidine bound here. Comparatively, in the green spectrum at 7.4, this region is more intense. And so we think that we have 2 histidines bound. The pH9 spectrum was a little bit trickier because at sort of first glance, looking at the intensity, it almost looks like we have about 1.5 histidines, which is entirely possible because we think that these are could be a mixture. So we're fitting kind of the average coordination environment. But we also found a really good fit by bending these histidine residues up out of the plane by about 20 degrees. In the pH9, it's a little difficult to see in this particular figure, but this first shell is actually more intense than in the other 2 spectra, and so we hypothesize that this may actually be a 5-coordinate species. And so with that, I'll pause for a moment if anyone has any questions on that first section.
There are several questions. Yulia, let's start with you.
Yeah, hello, nice talk. And my question is actually very early in your talk to your scheme of synapse, if you can kind of get back to it. Just a clarification question.
Sure, I can try. Let's see.
Yes, this is, so you see here you have MT3, metallothionein 3, and it looks like by errors, I don't know what to make out of those errors, but it looks like it's coming from astrocyte, but astrocytes, they express MT1 and MT2, but to the best of my knowledge, they should not express MT3. So I want your comment on, and why it is MT3, not other 1 or 2?
That's a good question. I will not take full responsibility for this schematic. This is one that I had borrowed from an earlier paper, and actually it's not something that had jumped out at me, but that is a fantastic point, that it is interesting why they chose MT3. And I thought it was also a little strange that it shows it almost just the way that the arrows are kind of drawn here. So I would guess that you're most likely right, that this should actually be one of the more common like brain metallothionein. So thank you for pointing that out.
MT3 is supposed to be expressed in neurons because like synapses are between neurons. So it's just a question. Okay, thank you.
Just, yeah, no, thank you. Good point. Thank you.
Okay. Marcin, you have a question?
Okay, thank you for a nice talk. My question is a little bit more technical. You said about HERFD and the problem with the photo reduction of your samples, but for HERFD you need a small beam and long acquisition time. So what is the real acquisition time and beam size for these measurements?
For our measurements, we're using a fairly large beam. I believe it was about 0.5 millimeter in height and 3 millimeters wide, might be 1/4 millimeter high, but this is on SSRL beamline 7.3, so it's a relatively low flux beamline. There's minimal upstream in terms of like focusing optics, so we can have that larger kind of blurred beam, which works really well for this type of sample.
And then how long did one spectrum of HERFD takes?
It kind of depended on which ones we're talking about. I think in the
amyloid, I guess.
In the beta amyloid one, where we were looking at the photo reduction, I believe those scans were about 30 minutes. We can go faster in terms of scanning in other samples where occasionally the rate of photo reduction is affected by a number of different components, and so we can scan faster. But it kind of becomes a question of you're getting the best signal to noise and minimizing photo reduction.
And using jet instead of frozen sample would help to do it on solution?
We are at liquid helium temperature, so we're at about 10 Kelvin typically.
Okay, thank you.
Thank you.
Hey, Kelly, to clarify, you had 2 different measurements. You had the ordinary fluorescence mode measurement and you had the HERFD.
Yes.
What was your, did you have to go to a smaller spot size for the HERFD?
Yes, we
And-
did.
Okay, and how did you check for damage in that case?
We did the same thing. Luckily, in the HERFD spectra, we're just collecting the near edge region. So those scans are only a couple of minutes long. And so we're using a very small beam. And so we were able actually to collect at even more points across the sample.
Okay. Oh,
so
good point. So you just have to
think-
raster more. Okay. Okay.
Yes, yes.
All right. Good. Next question is I lost. Oh, Yang Ha, you have a question?
Yes. So in your DFT models, you show the like energy level with different kind of like binding motif in your
Sure.
talk.
I can.
Yeah.
Let me just.
So I realized that some of the oxygen atoms have different protonation states. So I wonder if that is a fair comparison in terms of energy. Look, they're off by like 300 kilojoules per mole. So that's a lot. I wonder if that's from the protonation states. And also your data showed like different binding models at a different pH. And I wonder if that is consistent with the pKa of these tyrosines or phenolates.
So for this one, we're looking at small molecules. These are some of the 8-hydroxyquinolines. And in these calculations, we did account for differences in protonation state, differences in charge. I don't want to go into so much of the details here at the moment, but I mean, I'm happy to go into some of the details of how we actually did that calculation, but we did correct for those changes. In terms of the pH, we're talking about.
So yeah, so are they consistent with the pKa of the ligands, basically?
If the changes are consistent with the pK?
That's right.
It could be related, but we're specifically looking at here at the ligands that are bound to copper. I suppose changes in it could be affected in that way by the pH, depending on what is protonated. I think I get where you're coming at, but in this case, we're just looking at what is bound. So it could.
Have you considered any possibility that solvent water can bind, or you have all your ligands from your peptide or your protein?
That's a good point, and that's actually something that's been kind of debated in the literature, is whether or not water might be contributing to the coordination sphere. Unfortunately, with our data, we can't really tell where those other oxygen-nitrogen ligands are coming from. So it's based on our data alone, it's possible that it could be from water. Based on some of the other literature data, I think the agreement generally is that all the ligands are coming from the peptide rather than from water.
Thank you.
Thank you.
All right, there's another question, but we're going to hold questions to let you keep going, so we're closer to our timeline.
Okay, let me just jump ahead here a little bit. And on we go to the next question, which was how do 8-hydroxyquinolines bind copper in solution? And so we looked at a number of different 8-hydroxyquinoline-based chelators. The two that I wanted to draw your attention to are clyoquinol, CQ, and PBT2. These ones actually showed the most promise as potential Alzheimer's therapeutics. And so these have been studied the most, I guess, within the literature. And in general, 8-hydroxyquinolines are thought to bind copper-2 in a 2-to-1 bis complex. The copper is coordinated through the oxygen and nitrogen ligands in an approximately square planar coordination geometry. And so when we set out to confirm this by XAS and XAS, you can see this is our conventional XAS spectrum in the dashed line, and our HERFD XAS in the solid line. And now most of these, what we're calling 5,7 dihalogenated 8-hydroxyquinolines, so ones like cliaquinol, actually this pink spectrum is cliaquinol, they have very distinctive XAS features. So they actually have a 1s to 4pz ligand to metal charge transfer shakedown transition. And I just want to point out that this is very distinct from the 1s to 4p that we see with copper 1. This transition is fairly well established to be indicative of the square planar coordination geometry. These complexes also have structure in the higher K region, this sort of pattern that is from multiple scattering through the ring system and the halogens. And they also have peaks in the Fourier transform that correspond to halogen substituents. So in cliaquinol, this is the iodine substituent and this is the chlorine substituent. And I should mention that it's fairly unusual to see peaks this far from your absorber, which in this case is the copper. And I'll go into a little bit about how and why we're able to see that in these cases. And when we ran the spectrum of copper-2 PBT2, we noticed right away that these spectra are very strikingly different. And so fairly easily concluded that PBT2 appears to be binding copper very differently than some of these other 8-hydroxyquinoline compounds. And so just to summarize it kind of visually, 8-hydroxyquinolines that kind of look like this, the more traditional, this is cliaquinol, bind copper as proposed in the literature in this bis complex. So you have 2 of these 8-hydroxyquinoline moieties. You have coordination through the oxygen and through the nitrogen. And this copper is effectively a square planar. Actually, this whole complex is very planar or flat. PBT2, on the other hand, binds copper in unknown way. And so our question of how do 8-hydroxyquinolines bind copper really turned into a question of, well, how does PBT2 bind copper in solution? And so we started with a dichlorinated version of 8-hydroxyquinoline, which is kind of arguably a precursor to PBT2. So it has 2 chlorine substituents in the 5 and 7 position. And you can see here, this first peak in the Fourier transform corresponds to angle scattering to the oxygen and nitrogen ligands, which appear at roughly the same distance. And you can see some multiple scattering to the carbons in the ring, kind of in this region. You see copper multiple scattering through the oxygen to the chlorine, which actually is positioned at almost 180 degrees to the copper. So it gives us sort of maximal multiple scattering. And we also see a small peak in the Fourier transform, which likely corresponds to copper multiple scattering through the ring out to this chlorine in the 5th position. And so to look into this just a little bit further, I know the slide looks really busy, but we'll sort of go through it piece by piece. So as I've mentioned here, in many of these compounds, the copper, oxygen, in this case is iodine, just because the peaks are much bigger, more distinct to see. This angle is really approaching 180 degrees. This is actually calculated to be about 179.9 degrees, which means that we get really great multiple scattering through this angle. In this case, the oxygen and the nitrogen distances are so similar we can't distinguish them by XF, so they appear as this first peak. And then this is the this oxygen, or this iodine here, and this peak corresponds to this one. And so you can see in our simulated data, we have sort of replicated this structure at higher K. You can see this is the path that corresponds to the copper oxygen and the copper nitrogen. You see that they're in phase. And then you can see these multiple scattering paths out to this iodine through the oxygen and the significant contribution that they have at this higher K. And you can also see the copper iodine, which is the fifth position here, also has some contribution to this higher K XF structure. And I've just omitted some of the peaks that correspond to carbon in the ring just for clarity. And so through building several different models and trying to figure out what's going on in PBT2 and others, we realized that we could rotate around this oxygen so that we're keeping this copper oxygen distance the same, but we're lengthening this copper nitrogen distance. And so this does several things. It means that these two distances are now distinguishable in our Fourier transform, so we have two different peaks. It also means that this path, this angle of this path is deviated from 180 degrees. So that also results in dampening and changes to these multiple scattering peaks here. And I think, there we are. So the oxygen and nitrogen paths, the multiple scattering, which you can see is somewhat dampened in both, to both the iodine residues. And these are actually now almost completely out of phase. And so we can get a rough idea of what might be happening in our PBT2. And so if we go back here and we look again, this is our dichlorinated 8-hydroxyquinoline that we looked at before, our copper to oxygen and nitrogen distance. There's the chlorine in the 7th position, the chlorine in the 5th position. And so when we looked at this, this is also a dichlorinated 8-hydroxyquinoline, except now we have a methyl in this 2 position. And this results in the complex being twisted. We've pulled one of the 8-hydroxyquinolines out so that they're no longer coplanar. And this is exactly what we see. We see a distinct or almost distinct copper oxygen, copper nitrogen bond distances. We see a slight change in multiple scattering to the chlorines. And then PBT2, which is has a much bulkier substituent in this 2 position, has a greater rotation up out of the plane. And so you can see that these almost have 2 distinct peaks here, as well as dampening of the multiple scattering to the chlorines. And to make things a bit more complicated, we realize that there is a possibility for different copper coordination within PBT2. This 2-position substituent could actually coordinate the copper rather than this oxygen. And so our conclusion was that there is most likely a mixture of these 2 species in solution. And then we went on to look at how 8-hydroxyquinolines interact with amyloid beta in solution. And so we started again by looking at cliaquinol. So we took a solution of copper-2 amyloid beta. This is also the full-length peptide, so we have all 42 residues. And then we added 2 molar equivalents of cliaquinol. And we found through fitting our near edge spectrum that about 83% of the copper is now bound to cliaquinol. So it sequesters about 83% of the copper. And about 17% is remained coordinated to amyloid beta. When we did the same thing with PBT2, we found that only about 49% of the copper could be pulled out of amyloid beta by PBT2. About 41% of the copper remains bound to the peptide. And actually, we're going to jump away from XAS for just a moment because it's a little bit easier to see what's going on when we did some EPR. Whoops. So we see here, this is our spectrum of copper-2 amyloid beta at pH 7.4. So you can see that it's clearly a mixture of species. But interestingly, and surprising to us at the time, when we added added PBT2 to amyloid beta, you can see that there's still a mixture of species, but maybe not as many as we initially started with. For comparison, this is our PBT2 spectrum. And so the spectrum of our mixture does not look identical to PBT2, so we could see fairly clearly that PBT2 hadn't sequestered all of the copper. And so we subtracted our mixture of amyloid beta, copper, and PBT2, subtracted PBT2 out of our mixture of amyloid beta plus PBT2, and we got this spectrum that looks essentially like one single species in solution. And so this led us to hypothesize that maybe PBT2 can only remove copper from one of the components or one coordination environment from amyloid beta. And we did some preliminary calculations. We've since repeated these for the paper that we have almost ready. But these are some of our preliminary data. And so you can see here, this is the PBT2. And this is the sort of backbone of amyloid beta. This blue dot is the copper 2. And I haven't shown cliaquinol, but basically cliaquinol has no problem slipping into this pocket and likely has much easier access to this copper than PBT2. This 2-position substituent kind of gets stuck on the outside of the peptide, and it's doesn't slip as readily into this pocket. So this is kind of our working hypothesis that there may be a couple of different conformations, one that's more accessible to PBT2. And with that, I'll pause again for a few more questions before we go into the last stretch.
Since we're only about 12 minutes or so left, I'm going to hold these questions until the end. You should continue.
Sure, that sounds good. Thank you. And so in the last part, we're going to change gears just a little bit and look at how do 8-hydroxyquinolines affect cells in culture, what are amyloid plaques composed of, and then working towards answering the question, how do 8-hydroxyquinolines affect the Alzheimer's disease brain? And so for these, we've used two imaging methods. We use X-ray fluorescence imaging, which is a fantastic technique for biological samples because it again requires very minimal sample prep. Most of my studies were done with tissue that was just flash frozen, no fixatives, and put on an X-ray friendly substrate, something like silicon nitride, and then X-ray fluorescence imaging can give us elemental composition as well as localization information. And I also coupled XFI with Fourier transform infrared or FTIR mapping. These are also great for biological samples because they also requires very minimal sample prep. In many cases, if you choose your substrate right, you can actually image using FTIR mapping first, and then go on to collect your XFI maps. And the FTIR gives you chemical composition and localization that can be really complementary to your XFI information. And so just to quickly go through X-ray, or I guess remind you about X-ray fluorescence imaging. So we're raster scanning our sample. In this case, I've just drawn a cartoon of a brain. And so we raster scan through your sample. At each point, we get a spectrum that looks something like this. And each of these peaks corresponds to an element. In this case, I've highlighted iron in red, copper in green, and zinc in blue, and we can pull out that information at each pixel. We can fit these peaks to get quantitative information. And we can do this with several different sizes of samples. We have done single cells, we've done tissue sections. This is a brain section from a mouse, and we've done a little bit of whole animal sections. So there's really a range of information that you can get. Similarly, with Fourier transform infrared mapping or FTIR mapping, you get a spectrum at each pixel, and you can isolate peaks of interest to give information on lipids, proteins, carbohydrates, even lactate and nucleic acids. And so for our first question, how do 8-hydroxyquinolines affect cells? We used an immortalized cell line, a brain cancer cell line, and we noticed that when we added our drug at relatively low concentrations, we noticed it didn't have a significant effect on the viability of cells compared to our controls. But when we co-treated with copper and drug, we noticed that there's a significant decrease in the viability of these cells. And so we looked at the copper concentration. And so there was very little change when we added just the drug, but when we co-treated with copper and 8-hydroxyquinoline, in this case, it's the dibromo version in green, cliquinol is in pink, and PBT2 is in blue, we noticed that the intracellular copper concentration went up significantly when we co-treated with copper. And so this data was really inconsistent with a simple chelation mechanism. And so we looked further by collecting our XFI maps. These are our control cells here. You can see in blue, zinc is in blue, and it's kind of throughout the cell. The iron is shown in red, and it kind of surrounds the nucleus. And the copper levels are fairly low in the control cell, just kind of just above our detection limit, really. But when we co-treat with cliquinol and 3 equivalents of copper, you can see that copper, which is shown in green, is now throughout the cell. In comparison, when we treat with PBT2 and 3 equivalents of copper, you can see that zinc is still throughout the cell. We still have a similar pattern of iron near the nucleus, but the copper is localized kind of on the outside of the cell, possibly at the cell membrane. And so we had hypothesized that while the copper brought into the cell using cliquinol may be bioavailable and maybe causing the cell death, the PBT2 and copper may be getting trapped in the cell membrane, which is still causing problems, but maybe not as detrimental. And so for my last point, we looked at what are amyloid plaques composed of. And this is where we coupled FTIR mapping with XFI and found that amyloid plaques are actually a complex mixture of aggregated peptides. You can see those here in the FTIR maps, as well as lipids and metals. And each of these components have a very distinct distribution. So you can see kind of the plaque core well highlighted by the aggregated proteins. You can see that there are lipids kind of surrounding this aggregated protein core. And similarly with the metals, you can see that the zinc is really concentrated in the center or the core of the plaque. Iron kind of surrounds the zinc, and then copper is kind of distributed through overall the whole plaque region. And so with that, I will just quickly summarize what we've done and where we're headed with this project. So copper-2 binds amyloid beta in several different coordination environments. PBT2 binds copper-2 differently than other 8-hydroxyquinolines. And it may be due to this possibility for multiple coordination complexes in solution. PBT2 interacts differently with the amyloid beta peptide. PBT2 only sequesters a fraction of copper-2 from amyloid beta in comparison to other 8-hydroxyquinolines. And PBT2 interacts differently with single cells, so that the added copper appears to be associated with the cell membrane. And amyloid plaques appear to be a complex mixture of aggregated peptides, lipids, and metals. And so we are moving towards understanding the interactions of these 8-hydroxyquinolines in biological systems, specifically how they might interact with amyloid plaques within the brain. And so with that, I would just like to acknowledge all the people that have helped out with this project over the years. Of course, my supervisors, Ingrid Pickering, Graham George, and Stephen Urquhart at the University of Saskatchewan, a number of collaborators, Hugh Harris and his group synthesized a number of 8-hydroxyquinolines, especially when we kind of wanted to specify specific ones with different substitutions. Glenn Milhauser and his group synthesized amyloid beta, and Mark Hackett was a huge help when it came to the FTIR mapping. And of course, all of the group members, which I'm noticing are mostly past members of Graham and Ingrid's group. And then of course, all of the synchrotron facilities that made this work possible. We went to several to collect all of this data. And then of course, the funding agencies and all of you for coming and listening today. So thank you very much. And I'm happy to take your questions.
A very, very nice talk. A couple of questions from Svetlana Litsenko.
Hi, Kelly. So I really learn a lot. So it's very nice work, very well presented. So I have a couple of questions. So one was related to a beta study when you were looking at histidines binding coordination. Do you know whether or not these different forms you see are always coming from the same beta molecule, or it could be coordination between different A beta?
That's a good question, and it's actually been debated quite a bit in the literature, whether particularly with the amyloid beta 1 to 42, where it has this propensity to oligomerize and aggregate, that it could actually be coming from two different molecules. So it's possible. In our studies, we think that it's most likely coming from just one, because based on our concentrations and in solution, we had a surfactant in there to try and help minimize sort of protein-protein interactions, but it's certainly possible.
Right. So the other question was with hydroxyquinolines. So I find it really fascinating, the different distribution and different pattern. So one thought I have, so I assume if they're used for Alzheimer's, so they're blood-brain permeable, is that correct?
Yes, that's correct.
So was there any considerations for using them as a kappa chelator compound for kappa deficiency like Menkes, and if not, why?
I believe cliquinol has been used as a chelator in a number of different diseases over the years. It's been kind of a popular little compound because I think it's relatively easy to synthesize. I don't think it's actually been tested in Wilson's or Menkes, surprisingly. But
So I was curious because you clearly see very significant increase in kappa concentration when you use the complex, right? So when you use, and that's interesting because like by itself on their own, so they have significantly less effect. So I was sort of curious whether or not it works similarly to eclosamol, which people used recently for Menkes models, to actually deliver kappa across the blood-brain barrier into the kappa deficient brain.
it's a great idea. I don't think anything like that has kind of been tested, and there are relatively few studies that have looked at metal levels within the brain related to Alzheimer's. And I guess the studies that are out there typically don't have localization information. They're kind of from homogenized brain tissue to give like an overall look, because it was quite a long time before they information about the higher metal levels just within the plaques kind of reached the literature, and that it was not so much as overall the whole brain had severely altered metal levels, but it's really localized to the plaques. So it would be really interesting to do that experiment.
Yeah, excuse me, can I interject a little bit here? Svetlana, I think she showed that when she uses kappa inhalator, the cells are dying. Maybe that's not a viable delivery vehicle if it's toxic.
Well, it all depends on concentration, right? So that's, you're absolutely right. So that may not be very good delivery molecule if sensitivity is very high. but I don't know what concentration did you use, Kim?
They're relatively low. They're 5 micromolar of the drug. I should add in, I sort of glossed over it for the purpose of this talk and purpose of time, but the 8-hydroxyquinolines, particularly co-treated with copper, are really toxic to cancer cells. So they are toxic to immortalized cell lines in culture quite often, but when people have looked at them in vivo with healthy tissues, they're non-toxic on their own.
Very interesting. So last question. So do you know if PBT2, when it binds to plasma membrane, whether or not it's extracellular or intracellular?
Unfortunately not. We don't quite have the resolution to tell which side of the membrane it's on. And the purpose of using that dibrominated chelator was that we could track the localization of that chelator using the bromine tag. And when we were first doing those experiments, we didn't realize that the key one was going to be PBT2. So after the fact, we had really thought, really wished that we had a way to track where the PBT2 was, because we can see the copper, but not the chelator. And so we're sort of making the assumption that the PBT2 is still bound to copper, but it may not be.
Well, very cool. So I have more questions, but I'm not going to monopolize the time. So I'll see you later at home.
Great. There were great
Thank you.
questions. Last question for the seminar is from Yang Ha.
Yeah, you showed that you can have multiple binding site and actually a mixture when you bind. So can we conclude that? So in this case, we do not need to target a specific binding site at the protein, but instead just some chelating effect is just enough to treat the disease.
Possibly. It's interesting that they saw promising effects with cliquinol treatment and PBT2. So it's a little bit unclear exactly what the best mechanism is for removing the metals. And we'd actually talked a lot about kind of the working hypothesis and is it a good thing? Should we be pulling out all of the copper and solubilizing the amyloid beta? You know, what happens at that point if all of the amyloid beta from the plaques, excuse me, is suddenly sort of being degraded and you have this kind of big sort of bolus of soluble peptide trying to be cleared from the brain? What kind of potential is there for further problems down the line? Or would it be better to kind of try and design some kind of drug or chelator that might, for lack of a better word, kind of bind and inactivate the copper, potentially ameliorate some of that redox chemistry, but to leave the plaque in place? Because there are actually quite a bit of information in the literature that's saying that the plaques on their own may not be the problem. It may be, or at least amyloid beta might not be the problem, that it may be other things that get trapped within the plaques, either metals doing redox chemistry or other molecules, small molecules that get trapped. So unfortunately, I don't have like a very clear kind of answer to your question, but thank you.
Thank you.